Welcome from our side from Miltenyi Biotech. My name is Felix Eppler. I'm Global Product Manager for Cell Sorting at Miltenyi Biotech. And with me is my colleague Niloufar Monaseri. She's the Global Product Manager for tumor-infiltrating lymphocytes. And we're happy to discuss today how to harness the power of tumor-infiltrating lymphocytes for cellular therapies. So we will, before we go into a roundtable discussion, start with two presentations. Niloufar and me, we will kick off with a technical introduction to give you an idea about solutions and workflows we cover from Miltenyi Biotech side. And then Sandy Pelletier will take over to give an application example from the field and how the MACSQuant Tyto Cell Sorter helps her in her preclinical work utilizing tumor-infiltrating lymphocytes to fight melanoma. And with that, I would like to hand over to Niloufar as she will start with our technical introduction.
Thank you so much to Felix. Hello everyone, my name is Niloufar and today me and Felix would like to give you a brief introduction about the Miltenyi Biotech platform in TIL's clinical research field. We are at the forefront of pioneering clinical research with emphasis on immunotherapy, tissue regeneration, and graft engineering. Our validated products for cell manufacturing, such as Clinimax Prodigy platform and MACSQuant Tyto Cell Sorter, enable GMP-compliant cell processing and have been used in more than 50,000 cell therapy procedures. And also, we offer a broad range of training and services to our customers. For getting more information, please don't hesitate to contact us. Okay, let's start our main topic. TIL's clinical research with a special focus on the MACSQuant Tyto Cell Sorter. As you know, T cells are an important part of the adaptive immune system, and they play an essential role in fighting against cancer. T-cell adaptive cell therapy consists of CAR T-cell therapy, T-cell receptor therapy, and the last but not the least, tumor-infiltrating lymphocytes therapy. CAR T and TCR are genetically modified T cells which obtain a specificity to a certain antigen, whereas TILs are expanded tumor-specific T cells that exist in the patient body and in the contact to the tumor cells. Our main topic for today is clinical research TILs therapy, so let's go through that. TILs have proved promising results in treating certain solid tumors. TILs are known as a driver of personalized cell therapy because TILs can recognize neoantigens. Neoantigens are the antigens that are very specific for each patient because these antigens results from unique mutation acquired during cancer development. TILs therapy showed not only a very promising results in melanoma, but also other type of solid tumors such as breast, cervical, ovarian, and also colorectal cancer. But TILs therapy field faces some challenges. But today in our introduction, we would like to discuss the TILs manufacturing challenges and our solutions regarding GMP-compliant sorting of antigen-specific and most activated cytotoxic T cells from tumor tissue. Let's see how is the workflow for clinical grade TILs manufacturing and what are the challenges. In the conventional protocol, there are 4 steps. I've mentioned the challenges in orange, but because of lack of time, I cannot really go more into the details. It starts with collection of tumor tissue, which can be digested to get a single cell suspension or cut into fragments and grow in plates. Then isolated TILs should be activated and grow in a static phase. Afterwards, cells go to the dynamic phase, which is a rapid expansion. And at the end, final formulation. According to the personalized type of therapy, different type of tumors, stages, and so on, there is still no identified cell number or protocol for this step. So, finally, cells are infused to the patient. In summary, in conventional approach, there are lots of hands-in, which for sure needs very well-trained personnel. In addition, cell production requires GMP-grade facility, and what is obviously missed in the conventional approach is the specificity in the sense of selection of most activated killer T cells. If we have any solution for you, the answer is yes. At Miltenyi, we offer wide variety of GMP products in addition to CliniMax Prodigy platform and MACSQuant Tyto Cell Sorter, which guarantee your clinical grade TILs manufacturing. So let's start to have a look at the technologies that we provide you. CliniMax Prodigy. The CliniMax Prodigy represents the next generation in automated cell processing. The device offers advanced integrated solutions to streamline cell processing workflows from cell separation through cell culture to formulation of the final products. Regarding to TILs manufacturing, very soon we will provide you a comprehensive CliniMax Prodigy process which will bring novelty, automation, and reproducibility in a closed single system for you. Now I would like to ask Felix to go ahead with more information about Tyto cell sorters and the technologies behind.
Yeah, thank you very much, Niloufar, for that very interesting introduction into tumor-infiltrating lymphocytes and also some workflow solutions and technology solutions we have from Miltenyi Biotech. And now it's my pleasure to introduce to you the MACSQuant Tyto Cell Sorter. The Tyto, in contrast to the CliniMax Prodigy, works based on fluorescence signals and can detect multiple parameters at the same time. So the Tyto comes with 3 different lasers and can detect up to 8 fluorescence signals and 4 scatter signals at the same time. As you can see on the left-hand side, the MACSQuant Tyto is a small benchtop-sized cell sorter. There are no external fluidic tanks you have to store somewhere in your laboratory. You can control sample temperature between 4 and 25 degrees of Celsius, and you sort your cells within a closed and sterile environment, the MACSQuant Tyto cartridge. So let's have a closer look at the MACSQuant Tyto cartridge. On the left-hand side, you can see that we distinguish 3 different compartments inside the cartridge. There's an input chamber where we put the sample of interest in, a positive collection chamber where all the target cells are collected, and a negative collection chamber where all the rest simply flows through. Please note, there's no waste in the MACSQuant Tyto cartridge, meaning that we won't lose any of the precious sample material coming from the patient. All cells can retrieve from the cartridge all time. If we look at the bottom of the cartridge, we see that there is a microchip implemented, and that is actually the area in the cartridge where the analysis as well as the sorting takes place. So let's have a closer look at this area here. If we magnify this microchip, we can see that it consists of different sorting channels. Coming from the input chamber, the cells will enter this microchip on the left-hand side, run through the sorting channel and pass the different lasers. And once they are recognized as target cells, they will be very gently redirected into the upper compartment and then end up in the positive collection chamber. But how is cell sorting in general mediated in the MACSQuant Tyto? Let's have a closer look. So the sorting process itself is mediated via a microvalve, which is opened magnetically. An opening and closing cycle is really, really fast and done in 30 microseconds. As you can see here, once a target cell is identified, the valve opens and very gently and very fast redirects the cell into the upper sorting channel, which then leads to the positive collection chamber. So what are the benefits of microchip-mediated cell sorting? Well, first of all, it's very gentle. It runs at low pressure, so no decompression is acting on the cells. Also, since the sorting process itself is mediated via a microvalve, no charging of the cells is required. And also the shear forces acting on the cells are very mild, resulting in highly functional and viable cells, which are really important to be maintained for cellular therapies, as the cells still have a job to do. Furthermore, the system is quite easy to use and the cartridge serves in a plug-and-play fashion. But also depending safety, the MACSQuant Tyto Cell Sorter brings some benefits. First of all, the sample is all the time maintained in a closed and sterile surrounding of the MACSQuant Tyto cartridge. So there's no sample-to-sample carryover possible, and no contamination is coming into the sample from the outside. Furthermore, the microvalve-mediated cell sorting does not produce any droplets or aerosols, so it's also safe for the operator. And as mentioned before, sample is never lost inside the system, since the cells won't ever leave the cartridge. The only thing coming in and getting out is sterile filtered air. So why is the MACSQuant Tyto a good fit for clinical settings? As mentioned before, sorting happens exclusively in the fully closed and sterile cartridge. There are no fluidics inside the instrument itself. Furthermore, all consumables are available in Max GMP grade, which holds true for our Tyto cartridge, the Max GMP Tyto running buffer, as well as a portfolio of fluorescent antibodies. This means that everything in direct contact with the cells comes with an extensive documentation. Currently, the MACSQuant Tyto Cell Sorter is approved for 10 different clinical trials, either in phase 1 or phase 2, and over 200 patient samples have been sorted to date. So if we look at application areas where the MACSQuant Tyto Cell Sorter is used in clinical settings, you can see that we have quite a variety on cell types being sorted with the MACSQuant Tyto, reaching from fields like immuno-oncology to regenerative medicine. In the following, I would like to give you 2 brief examples on applications. The first one is coming from Japan, from the Center of iPS Cell Research and Application, where the group of Jun Takahashi leads a pioneering clinical trial, and they utilize the MACSQuant Tyto Cell Sorter in order to enrich dopaminergic progenitor cells to high purities. Once the differentiation protocol is complete, those cells are utilized in order to treat patients suffering from Parkinson's disease. If you're interested in more details on this, you can find a poster being published by the group on our website. Please just follow the link. But coming back to immune oncology, we also see some nice applications with the MACSQuant Tyto Cell Sorter. For example, we have quite some clinical trials currently running where the MACSQuant Tyto Cell Sorter is utilized in order to enrich antigen-specific T cells directed towards different kinds of cancers. If you're interested in more examples and more details about that, please find those webinars which also were held here on Cell and Gene Therapy Insights, either on this website here or on our Miltenyi Biotech website, and look for more concrete examples on how customers from, for example, Emetics, as well as the MD Anderson Cancer Center, utilize the MACSQuant Tyto Cell Sorter in order to work with antigen-specific T cells in clinical trials. And with that, I would like to hand over again to Nilüfer, as she would shortly like to wrap up our technical introduction.
Thank you so much, Felix, for a very nice overview on MACSQuant Tyto Cell Sorter and its great application in immuno-oncology and also regenerative medicine. But I have one last point. Please don't forget, for being successful in your future clinical trial authorization, you need to have a precise plan and prediction. What we all know is that when you are in a preclinical stage of development, you can easily produce a complex product and are quite flexible. When you enter into a clinical trial, your process becomes inflexible while your patient's cohort increases. The patient cohort continues to increase during later clinical trial phases until market access, and you need to start upscaling. So this means looking very well and accurately at technologies or suppliers that are able to help you to meet your future needs. Looking for technologies and products that can be used for preclinical research or process development to clinical trial is important and very critical for your future success. Thank you so much all for your attention.
Thank you very much for that short wrap-up, Nilüfer. And with that, we are happy to hand over to Sandy, and we are already eager to learn more about her application in the preclinical stage currently. Thank you very much for your attention.
Hi everyone. So the title of my talk is Harnessing the Power of Tumor-Infiltrating Lymphocytes for Cell Therapy. I will talk about what is tumor-infiltrating lymphocyte adoptive cell transfer. I will talk about the features of the MACSQuant Tyto selection of cell surface markers for sorting and flow cytometry antibodies. I will also talk about how to prepare your cell suspension from solid tissue, and finally how to optimize your cell sorting purity and efficiency. So TillAct starts after a tumor excision. The TILs are expanded in vitro, and then they are infused back into a lymphodepleted patient along with IL-2. So several studies have looked at TillAct efficacy. So as we can see here, the objective response rates vary between 36 and 47%, while the complete response rate vary between 3 and 17%. So our team has focused on how we can improve these rates. So the challenges associated with low response rates to TillAct include the low frequency of tumor-reactive TILs in the cell infusion product because they are often overgrown by standard T cells in bulk expansion. And second, there is no good selection method for tumor-reactive TILs. So our approach is to make a cell product enriched in tumor-reactive TILs using X cell surface marker as a closed system and a closed system cell sorter. So for the purpose of the talk, I will call our cell surface marker X. So the literature has shown that sorting of X positive TILs in tumors can enrich in functional and tumor-reactive T cells. So if we look at the graph on the left, it is the percentage of tumor-reactive T cells after co-culture with an autologous tumor cell line. So we're looking at the upregulation of 4-1BB. So we can see that total CD8 TILs and the X-negative TILs express low levels of 4-1BB. However, the X-positive cells upregulate 4-1BB to a much higher extent. And on the right, we can see that these cells also secrete high amounts of interferon gamma when co-cultured with autologous tumor cell lines. So our approach is after tumor excision to perform transformation of the tumor into a cell suspension, and then to select X positive TILs with the MACSQuant Tyto Cell Sorter. Then to expand these TILs and infuse them back into a lymphodepleted patient. So the features of the MACSQuant Tyto. So if your goal is to sort cells and infuse them into a patient, the most important thing is to keep your cell infusion product sterile to ensure patient safety. So in common cell sorters, the cells pass through many tubing at high pressure and often at room temperature. So the MACSQuant Tyto offers a closed system. So the cell sorting is performed in a sterile disposable cartridge with low pressure, and the chamber can also be cooled. So here is a picture of our team and the Tyto in our clean room at the shim. So we also chose the MACSQuant Tyto because of the services that are offered by Miltenyi. They offer a contract service, of course, biannual maintenance. They also offer installation and operational qualifications. They offer live support. And the other advantages that they offer cartridges and sorting buffers in GMP grade, but they also offer them in research grade. So it's practical to use the research grade for starting the optimization process and then to switch to GMP grade later on. So as I mentioned before, the cell sorting is performed in one cartridge. So the cells are first added to the input chamber. They pass through a red, blue, and violet laser. So when a cell of interest is detected, the valve opens to let the cells of interest move into the positive collection chamber, and then the cells that are not of interest move into the negative collection chamber. So I next wanted to talk to you about how to choose your cell surface markers and your flow cytometry antibodies. So this will come not as a surprise, but choosing a marker that is highly expressed will increase your sorting efficiency. So when I talk about highly expressed, I mean in terms of mean fluorescence intensity. I'm not talking about the frequency of these cells. Also, we found that using the CD45 to distinguish immune cells from non-immune cells, such as tumor cells, was also very helpful. You have to keep in mind that working with tumors is not the same as working with PBMCs. It's not a homogeneous cell population. And also with the Tyto, we find that the side scatter and back scatter is not perfect, so it is sometimes hard to distinguish the immune cells from the debris, the dead cells. So CD45 was very helpful for us. The other thing that was helpful is adding a dump channel to exclude the undesirable cells instead of using a viability dye. So sometimes dead cells, debris, but also large tumor cells can sometimes bind non-specifically to antibodies. So by using a dump channel, this excludes these, this type of problem. So next, which antibodies to choose? So if your antibodies, if your markers of interest are available as GMP grade, that's great. And if there are very few antibodies available on the market, so another option that we thought was interesting. So if your antibodies, if your markers of interest are available as GMP grade, that's great. Unfortunately, there are very few antibodies available on the market as GMP grade. So another option that we thought was interesting is the area affinity antibodies from Miltenyi. They are recombinant antibodies. They have high purity, high lot-to-lot consistency, and they are animal-free, which is very important for regulatory agencies. And also, they do not have any, they have a mutation in the Fc portion of the antibodies, so Fc receptor blocking is not necessary. The other option is GMP-RUO or ASR grade antibodies. These are used for diagnostic purposes. They come as a lyophilized antibody, so they're very stable at room temperature. And what's practical is that some of the companies can even pool all of your antibodies in one vial, so all you need to do is add your cells to this vial. So this decreases the risk of, pipetting errors. And finally, if your antibodies, if your markers are not available in these formats, and if you don't have a choice to use a regular, for example, a mouse anti-human antibody, I would suggest to plan for rodent viral testing to satisfy regulatory agencies. So the third part of my talk is about how to prepare your cell suspension from a solid tissue. So you can either use a mechanical digestion or an enzymatic digestion or a mix of both. We use the Gentle Max system, which is a closed system. As for enzymatic digestion, there are several enzymes available, either collagenase, DNase, protease, and the enzymes that are needed will really depend on the type of tissue you want to digest. There are a few GMP or clinical grade enzymes on the market, but there are a couple. And the other thing I would suggest is to perform optimization. You need to think that these enzymes could affect the expression of your cell surface markers. So if your cell surface markers are degraded by the enzymes, you will not be able to sort your cells afterwards. And it's also important to look at cell viability and cell function when you put your cells in contact with these enzymes. And finally, if you've ever digested food before, you might have seen this type of dot plot. So as you can see in this example, the lymphocytes only represent 0.037% of all events. The rest are debris and dead cells. This can be the case, especially if your tumor was necrotic, if your patient has received chemotherapy before the surgery, you can get this type of sample. And this type of sample will definitely clog your cell sorter or the Tyto cartridge. So what I would recommend is to phycol to eliminate the debris if you need to. And phycol is also practical to eliminate the red blood cells, especially if you need to. And phycol is also practical to eliminate the red blood cells, especially if you have tumors that are highly vascularized. You can get a lot of red blood cells in your sample. And finally, beware of what we call mucinous tumors. So we've encountered this with some colorectal cancers. The mucin will clog your cartridge or any cell sorter. So really, you have to take this into account to know what could be the issues depending on the type of tumor or type of tissue you want to sort. So the last thing I wanted to talk to you about is how to optimize your cell sorting purity and efficiency. So basically, you need to know what is your ultimate goal. So is your goal to obtain 99% purity, or is 90% okay? So basically, is your goal to enrich or to purify your cell population? And second, the sorting efficiency and depletion yield. So, So for example, if your cells of interest are at a very low frequency, your goal might be to sort all of your cells and to have none of your cells end up in the negative fraction. So to show you what's the difference between the purity, the efficiency, and the depletion yield, I've added here an example. So this is a melanoma lung metastasis. So on the left dot plots, we can see forward and side scatter profile. On the left dot plots are the CD3 marker and our X marker of interest. On top are the pre-sort cells. In the middle are the post-sort positive fraction. And at the bottom, we can see the post-sort negative fraction. So in this example, the post-sort purity was 92.5%. The efficiency was 67%. So what's the efficiency? So basically, it's the ability of your sorting to sort all of your cells of interest and not have any of those cells going to the negative fraction. So it's calculated based on the number of your cells of interest in the positive fraction divided by the number of your cells in the positive fraction and the negative fraction. And finally, the depletion yields. It tells you how well you were able to deplete your pre-sort fraction. So it is calculated by deplete your pre-sort fraction. So it is calculated based on the percentage of your cells of interest in the pre-sort fraction minus the percentage in the negative fraction divided by the percentage in your pre-sort fraction. So in this example, we started with 36% of cells having the marker of interest. After sorting, we still had 12% of cells in the negative fraction. So in this example, we have a depletion yield of 67%. Some of the options to increase the purity and efficiency would be to play around with the cell concentration. This will definitely influence the abort rate and your overall purity. And the other option is to sort the negative fraction. So to perform 2 consecutive sorts, this way you'll be able to get all of your cells of interest that initially went into the negative fraction. So this will definitely increase your efficiency. So here's a summary of sort results. So on the left is the yield of total sorted cells in melanoma tumor cell suspensions. So it's highly variable. In some samples, we got a couple of thousand cells, and in other samples, we had a couple of million cells. So this will be influenced by your starting material, obviously, and also the immune infiltration in your tissue as well. In the middle graph, we can see the cell sorting performance. So in this plot, we have melanoma in full dots, and we also have GI cancers in the open circles. So the purity, we get on average around above 90%. In terms of viability, we always get above 95%. The efficiency and the depletion yield, we get on average 75%. And the ones that are on the lower end are sorts where we did not perform a second sort to get all of the cells. And finally, on the right, we looked at cell stability after sorting. So we measured the viability of the cells for up to 6 hours, and we've seen that in these 2 patients, the cell viability remains stable. So in this slide is a summary of T cell functionality after sorting. So on the left, we can see that there's excellent post-sort T cell fold expansion after 2 weeks of culture. So we get on average a 5,000 fold expansion of our T cells, although it varies from tumor to tumor, obviously. And we've also measured the functionality of our cells. So on the top dot plot are our sorted TILs with no target. On the bottom are sorted TILs that were co-cultured with an autologous tumor cell line. So we can see that the CD4 cells specifically upregulated CD134, which is also called OX40. And the CD8 T cells specifically upregulated CD137, which is also called 4-1BB. And at the bottom, we can see that the X-positive cells secrete interferon gamma when they are co-cultured with the autologous tumor cell line, whereas the X-negative cells, or the bulk cells, secreted much less interferon gamma. So in conclusion, our goal is to increase clinical responses to TIL-ACT using tumor-reactive TIL enrichment directly from tumors by flow cytometry cell sorting. We have optimized standard operating procedures to enrich tumor-reactive TILs with good purity, with good efficiency, very good viability and functionality. We have done our preclinical trial application meeting with Health Canada last September, and we hope to file the final CTA submission later during this year. So I would like to acknowledge my supervisors, Simon Turcotte, the entire Turcotte lab, also the people that are involved in the biobanking, le soutien de la recherche clinique, and all of the funding agencies.